pleasure to introduce and to welcome Joy Ward, Dean's Professor of the College of Liberal Arts and Sciences from the University of Kansas in the Department of Ecology and Evolutionary Biology. I know a number of you are colleagues of Joy. I've seen nice welcome. The big question that Dr. Ward is going to address is, can the past inform how plants will respond to future climate change?
Thank you. And thank you for bearing with us on the technical difficulties. Let's see. That's right. I'd like to first begin by pointing out that the planet is currently undergoing a series of major global changes. And obviously the one that we hear most about, particularly in the media, is climate change. And depending on the model that you're using and the CO2 emission scenario, the planet is expected to warm anywhere between 2 and 8 degrees Celsius by the year 2100. And that's a result of increases in greenhouse gases like carbon dioxide that trap long-wave radiation and reduce the loss of heat to space. Now, if we can assume a conservative estimate of warming at about 3.5 degrees Celsius, other models indicate that between 15 and 37% of current species will eventually be destined to extinction as a result of our activities on this planet. And therefore, that type of extinction event is rivaling some of the major extinction events that have occurred over the last 600 million years in the geologic record, although in this case, they are mainly driven by human activities. Along with that, other models predict that between 1/3 and 1/2 of the surface of the planet has been permanently altered through land use change. And this generally comes in the form of natural ecosystems being converted to large agricultural centers and urban areas. And obviously, those types of activities are critical for our economic growth, as well as for us to feed ourselves as a human population in agriculture. But at the same time, we need to realize that cycles such as the water cycle, the nutrient cycle, and the carbon cycle are being altered through those land use activities. And therefore, it's not surprising that the totality of all of these global change factors are having a large societal impact. So at this point, we're losing resources in the area of cultural as well as natural resources that are critical for our functioning as a human race, as well as other portions of ecosystems. And this is having implications both near and far. So in the work that I do in the Middle East and other areas, we're seeing very large reductions in water supply in those land regions. But these effects also come close to home, where, for example, in 2012, in that extreme year, we had a complete decline in our corn crop in the state of Kansas. And so these are having implications for populations far away as far as us and here in the Midwestern part of the U.S. Now, in the scientific realm, it is key that we understand plant responses to climate change in particular. And there are a number of reasons why that's critical, but I'll list 3 of those here. To begin with, plants capture energy from the sun, and they convert that energy into chemical energy. And therefore, they serve as the conduit to which energy from the sun eventually enters into ecosystems. And in addition to that, because plants can manufacture their own sugars, their own carbohydrates, they serve as the foundation of the food web on which all other aspects of the ecosystem depend. And then lastly, plants take up carbon dioxide in photosynthesis, and therefore they serve as a sink for the CO2 that we currently emit into the atmosphere. And in that sense, they can help mitigate some of the CO2 emissions that we're putting into the atmosphere. So those are a number of reasons plant research is critical in the realm of climate change. There are a number of different approaches to plant research and climate change, and they involve different time scales and different scales in the ecosystem. For example, some researchers focus at the gene level or the molecular level, and this spans all the way up to the ecosystem scale. And certainly in this room, as well as throughout the University of Minnesota, we have major leaders in understanding ecosystem level responses to climate change. Along with that, there's different locations where this work is being done. And the reasons for that are that plants that live in the tropics respond very differently to climate change factors than those in the Arctic, for example. And then lastly, and the one that we will focus on today, is that you can use different time periods and plant responses and adaptations to different climates and different CO2 levels of the past to possibly get insights into how plants may respond into the future. But I think you'll see throughout the talk that there are many times novel scenarios that result in plants adapting to interesting factors in the ecosystem, not only climate, and we'll look at those throughout the talk. So we will focus on a number of different time scales. The first will be deep time, and there we'll look at plant adaptations to an interesting novel factor in the environment, and this is at the time scale of hundreds of millions of years ago. The second case that we will look at is the last glacial period, which is tens of thousands of years ago. And there we'll look at survival of plants under interesting physiological constraints that relate to low carbon dioxide, which forms the basis for much of my research. And then lastly, we'll look at pre-industrial to present, and we'll look at how plants have changed in the last 200 years since the onset of the Industrial Revolution. And since that time, carbon dioxide has risen as well as temperature. And it's key that we understand plant responses to conditions prior to our effects, because that serves as a baseline to how plants would respond without humans. And then we can, from there, quantify the changes that we have invoked on plants because of our activities on the planet. And in addition to that, in studying this, we can then look into the future to avoid some surprises in how plants might respond into the future and know those in advance and prepare for those. So before getting into looking back into time, let's first take a look at the modern carbon cycle shown here. And this represents a flux. This is 9 gigatons of carbon per year, or in other words, 9 times 10 to the 15 tons of carbon that are emitted into the atmosphere each year by human activities, like the combustion of fossil fuels and land use change, which mainly comes in the form of deforestation. Now, not all of that carbon dioxide accumulates in our atmosphere, fortunately. There are 2 main sinks that take up that CO2 that we emit, or at least a portion of it. 2 gigatons of carbon that we emit are taken up by oceans, and approximately 3 gigatons of carbon per year are taken up by terrestrial vegetation. And therefore, it's absolutely key that we understand how long vegetation will be a sink to take up rising carbon dioxide, and what the physiology of plants might be in the future as we move forward. So rather than 9 gigatons of carbon being emitted every year, only 4 accumulate in the atmosphere each year, and that number actually is getting larger and larger as we continue emissions into the future. At least that's the predictions. As a result of that CO2 emission level, you can see in the yellow line that CO2 has risen from about 290 parts per million during the pre-industrial period to a current value that has now topped 400 parts per million in the last year or so. And along with that increase in greenhouse gases, we're also seeing a rise in temperature of about 1.5 degrees Fahrenheit, or about 0.85 degrees Celsius, since weather records began in 1880. And myself, along with the majority of scientists, believe that that temperature rise is a direct result of this increase in this level of greenhouse gas of CO2. Now let's look back into time. This is going to be at a time scale of about 250 million years ago, so way into the Earth's deep past. And what I'll be doing is showing you 2 tree specimens from time periods that are approximately 250 million years old. And during that time, notice that carbon dioxide was much higher than it is today. It was anywhere between twice or 5 times higher than it is now. And temperatures were also much higher on the Earth during that time period. So this represents an analog of the past where we can look back at a warmer condition in the environment to see what vegetation was like, or maybe where it was dispersed throughout the planet. At about this time, at about 255 million years ago, this is the distribution of the continents. They were in a supercontinent known as Pangaea. And the samples that we will be looking at are from Antarctica. And during that time, the plants that grow in these kind of regions today. However, when we think forward into the future, at about 2070 to 2100, we are expecting that Arctic regions will be about 8 degrees Celsius, maybe even higher, warmer than they are at present. And in response to that, we are beginning to see how a highly productive forest, how it was adapted to these very different novel environments of long-term darkness versus long-term light of the past, to give us insight into what trees may be faring with in the future. So looking back in deep time in our first example, we see that with climate change, with redistributions of plants as a result of climate, they can encounter novel environments like months and months of light and dark periods that help us understand what trees in the future may face. Now, in the next part of the talk, I want to look back tens of thousands of years to the last glacial period, a time when it was much colder than at present. And there we will look at an interesting physiological constraint of plant physiology. So during the last glacial maximum, we're moving way up in time, much closer to the present. This was only about 20,000 years ago. And during that period, these very large megafauna inhabited North America. And all of these megafauna went extinct as conditions began to warm, getting closer to the interglacial period. Now, the plants that lived during this time period were quite similar than they are today, although they were distributed across the planet very differently. So here you can see these ice sheets here shown in gray, and many of the boreal and temperate forests have been distributed farther south. And we know that from modeling efforts where we can recreate the conditions that they occur in now and advance those farther south during the last glacial period to get a sense of how they may have been distributed in the past. We also have independent clarification on where these trees have occurred through a really unique collection known as pack rat middens. And I curate one of these collections in my lab. And basically what happens are pack rats, if you know those rats, they go out and they collect trees from their local environment and other plants, and they bring them back into their dwellings. And in caves, these dwellings are very, very well preserved, particularly in the Southwest. So we have a real direct record over the last 20,000 years of the kinds of vegetation that occurred in a given area and how it changed through time, from the cold period of the glacial to the warmer interglacial of the current time period. So just to give you an idea, during the last glacial period, we had ice covering much of the northern part of North America. It certainly was covering this state in the past, and then all of those ecosystems distributed farther south. Okay, now in addition to the colder periods and the redistribution of vegetation, there was another factor that vegetation would have been dealing with globally, and this is low carbon dioxide levels. So during the last glacial period, globally, no matter where plants occurred, they were exposed to carbon dioxide levels in the atmosphere of about 180 parts per million. And we believe that during the last glacial period, this low carbon dioxide may have been limiting to plant growth, much like water or nutrients are today. And I can show you some examples of why we believe that. Before I do that, though, let me take one aside into photosynthesis just to show you where the carbon dioxide concentration might affect plant physiology. So as I mentioned earlier, plants are able to take the sun's energy and convert that to chemical energy to make carbohydrate with. And that assimilation of light occurs in the light reactions of photosynthesis. The assimilation of carbon dioxide occurs in the Calvin cycle, and all of these reactions occur in the chloroplast within plant cells. Now, the carbon dioxide that is fixed in plants and that is used to make sugars originally comes from the environment. It diffuses through pores on the leaves, known as stomata, and then it eventually enters the chloroplast inside of plant cells. And there, carbon dioxide is fixed in a reaction that is catalyzed by rubisco. And then eventually sugars are formed for the plant to use as a food source. So it's not surprising that if carbon dioxide was very low in the atmosphere, that this might limit photosynthesis and have large effects on plant growth. And when we take modern seeds of modern plants and when we grow them at low carbon dioxide conditions of the last glacial period, that is exactly what we see. So these plants have 90% growth when grown at the glacial CO2 level compared with plants grown at the current CO2 concentration. Now, you probably also have noticed that plants have higher growth at very high carbon dioxide. That's predicted for 50 years into the future. So it's a positive effect, but I want to point out that in these experiments, we are not exposing plants to high heat, droughts, and many of the other factors that will come with climate change that will actually reduce growth in the future. This is an isolated CO2 effect only. Also, in other work, we've taken all the work done with low CO2 in plants and put that together in these graphs shown here. The top is photosynthesis, and the bottom is biomass or growth. And you can see that a value of 1 is the standard for plants grown at the current CO2 level. And as we move back to the low CO2 of the last glacial, we see that photosynthesis and biomass production are reduced by at least 50% in a variety of species. And each one of these symbols represents a different kind of species. So these include annuals and trees as well. So low CO2 is a novel factor in the environment that was global during the last glacial period and would have had implications for plant physiology and plant growth. Now, all of that work was conducted with modern plants grown at past conditions. And it's not showing us how plants might respond that had thousands and thousands of years to adapt and evolve under low CO2 conditions. And in order to find answers to that question, we actually use ancient plants for this work. And in my case, we use plants from the La Brea tar pits that currently are in Los Angeles today. And the plants that are removed from there are mainly wood samples. They're incredibly well preserved in their original organic state. And you can even see the wonderful tree rings on all of the majority of wood samples that we collect. And these are about 20,000 years old on average. What we can do then is take the wood out of these plants and we extract cellulose from that wood. And we can run carbon isotope ratios on those wood samples to actually understand what the physiology of these ancient plants was like in the past. And I'll give you a quick aside on how we do that. So basically, in this room, for example, about 1% of the carbon dioxide molecules, which are available for plants, if a plant were to use one, have a carbon atom with one extra neutron on them. So we refer to this as carbon-13 CO2. The majority of CO2 comes in the well-known carbon-12 form. When plants are taking up carbon dioxide in photosynthesis, there are two factors that slow the uptake of C13 CO2. One is simply diffusion, where the carbon-13 CO2 molecule moves slower through the air and therefore enters into the plant leaf much slower than the carbon-12 CO2 form, and therefore less of it is taken up relative to the air. In addition to that, rubisco, which is that enzyme I mentioned that fixes carbon dioxide in photosynthesis, it strongly fractionates against carbon-13 CO2. And when stomata are wide open on the plant surface, it fractionates against C13 more. And so when we measure carbon-13 to carbon-12 ratios, we can actually use theory to get a value known as CI-CA. CI is the carbon dioxide concentration inside the leaf. CA is the carbon dioxide concentration outside of the leaf in the air. So basically, we're getting a sense of what the CO2 concentration is that is available to diffuse into plant cells and then to be taken up by rubisco in the chloroplasts relative to the atmosphere. And also, since we know the atmospheric CO2 concentration from ice core values, we can actually know what CI is, the intercellular CO2 level in these ancient plants. And that's critically important because if we know how stomata regulate the entry of carbon dioxide into plant leaves, we also have a sense of how much water loss is allowed as water leaves leaves. And this represents an important trade-off between carbon gain for photosynthesis and water loss through the same stomata on the leaf surface. So the data that I will be showing you next are for ancient specimens that lived during the last glacial period, and we'll be looking at what these intercellular CO2 concentrations are in ancient plants versus modern plants to understand the carbon dioxide resource availability in ancient versus modern plants. I will also add that on the x-axis is that intercellular CO2 level in leaves. This is plant growth on the y. And at low CO2 conditions, there are strong correlations between these intercellular CO2 levels and growth. So we can get some sense of what the growth rates were of our glacial plants based on these correlations with modern plants, if we allow for that. Okay, so this is the first set of samples. These are different juniper trees that lived during the last glacial period, and they're recovered from the La Brea tar pits in California. They range in age from 47.6 thousand years old to present. And I have two different kind of modern controls. The first control are low elevation juniper trees, which match up to very similar locations as the tar pits, but out of the immediate influence of pollution. They're going to have differences in CO2 levels, higher for today. Conditions are now drier in Southern California, and it's warmer. So there are a number of environmental factors that have changed. This is a high altitude control shown on your right. This is where we collect juniper specimens of the same species from higher altitudes in the LA area. But these trees are at cooler and wetter conditions, which recapitulate lowland conditions during the last glacial period. And so when we compare this group and this group, we're comparing similar climatic conditions other than these are at high CO2 and these are at low CO2. Now, each one of these lines represents one individual, and I stack the different tree ring values from the highest ones, the maximum, to the minimum. And this is the intercellular CO2 concentration that's available within the leaves of those plants for photosynthesis. And notice quite remarkably that CO2 concentrations inside their leaves are extremely low compared to modern vegetation. So these high CI values, these are tree rings or years in California when conditions were very wet. Stomata are wide open on the surface of the leaves, and they have high intercellular CO2 levels. Here you can see very dry years where their stomata are relatively more closed, and CI values go down because CO2 is used up within the leaf. All of our glacial trees are operating at CO2 conditions inside their leaves that are unprecedented, that are much lower than any of the vegetation that we have in modern ecosystems. So it's quite likely that these plants were starved for carbon during the last glacial period, which would have had large effects on their growth. Now that represents a response in the northern hemisphere. We've also looked at this in the southern hemisphere with kauri trees. These are from the North Island of New Zealand. These are giant trees, and they are removed from bogs in New Zealand, and they are used for the construction of sustainable furniture by a company known as Ancient Wood, and we use their specimens in our research. And these range in age from about 53,000 years before present to about 48,000. I've got the same kind of data here. These are the glacial trees. These are modern trees. And you can see yet again that the intercellular CO2 level of these trees is unprecedented compared with modern trees. So again, these are very low and probably have about half the growth rates if we just use the CO2 effect compared to modern trees. So we can see that during the last glacial period, climatic conditions were different. It was very cold, but there were also interesting novel physiological constraints globally where carbon dioxide levels were quite low, and trees may have been highly carbon starved during that period. Unlike what we're seeing today, where CO2 levels are rising and plants are having higher and higher CO2 availability. And I will add that adaptation to very long-term levels of low carbon dioxide over the last several million years are probably influencing the physiology of how plants will respond to CO2 as it rises in the future, since plants are more constrained and more conservative due to this adaptation to long-term low CO2. Okay, and to end here, I'd like to present just one more example looking back and how we can use past conditions to understand the future. And that's looking over the past 200 years, looking from the pre-industrial period to the present to help us establish a baseline for the direct effects that we've had in only a few hundred years. And as I mentioned earlier, CO2 levels were about 270 parts per million during the pre-industrial period relative to the current value of 400 and the level of about 700 that we expect to occur by the end of this century. So let's take a look at that research. Before showing you what we're working on, I want to show you a very classic study by Fitter and Fitter that was published in Science in 2002. And there they surveyed the flowering time of about 385 plants in England. And they compared their flowering times in the field during the cooler phase of 1954 to 1990 relative to the warmer period of 1991 to 2000. So cool versus warm period. A level of 0 corresponds to no change in flowering time. Positive values are delayed flowering, negative values are earlier flowering. And so what they found is basically over the last 50 years, the majority of the species surveyed are flowering much earlier in more recent decades relative to older decades. And many, many studies are in the literature now showing accelerations in flowering time. And the majority of interpretations involve the idea that this is due to warming. And certainly it is probably in many cases. But there were two factors that changed, if you remember, between the last glacial period, between the pre-industrial period and the present. Both carbon dioxide and temperature have risen. And in many studies, the impacts of rising CO2 are not really considered. So I'd like to take a look at that in the rest of the talk. I will also add that in a study done at Jasper Ridge, this is an excellent set of work at Stanford. There they actually emit a warming response for plants in the field. And in general, they have found that these warming experiments in the field actually underestimate the accelerations in flowering time that are occurring in the field. So these studies where only warming is imposed actually are not seeing the real accelerations that these kinds of field studies serve. And so it seems there may be a missing factor, and it possibly might be CO2. And so that's what we're looking at now. The system that we will be using to look at this is Arabidopsis. This is a genetic model system. It was also the first higher plant whose genome was sequenced, so we have a lot of information on this plant from the molecular all the way through the ecological level. And also notice that it responds very strongly to rising carbon dioxide with higher growth between glacial through future CO2 concentrations. However, in the next slide, we look at 6 genotypes, all of which also show higher growth between 350 and 700 parts per million CO2, but their flowering time responses are very different, so they're always larger at this treatment. But some actually flower earlier at higher carbon dioxide, and some of these flower much later at higher carbon dioxide. So that growth response is not highly predictive of what their developmental response will look like. And we're trying to weave apart the impacts of CO2 and climate on developmental timing. Also, this gives you an idea of what that CO2 effect looks like. This is one genotype of Arabidopsis, so this is the same genome in this plant versus this plant. This is grown at today's CO2 value. This is a value predicted for 50 years into the future, not very far into the future. And notice that this plant is about twice the size of this one by mass, but it's about another 2 to 3 weeks away from flowering. So high CO2 has had a really disruptive effect on developmental timing. So it may not just be warming, but also CO2 that will impact future plants. So to take a look at this, we wanted to disentangle the effects of warming and CO2 rise since the Industrial Revolution began. So I'll show you some new data from that work before concluding. So the way this will be organized is on the bottom will be the full pre-industrial period, which is low CO2 at 270, and the cooler pre-industrial conditions, the cooler temperatures. And on the Y will be any given experimental treatment. Now, if points fall along this 1-to-1 line, that indicates that their flowering time is the same between the full pre-industrial period and whatever experimental treatment is on the y-axis. If the data points are below, that means they're flowering much, much earlier. And if the data points are above, they're flowering much later. So let's take a look at what happens. And in this case, this is the full pre-industrial condition of 270 ppm CO2 and 20 degrees Celsius. And in this case, we're only going to change temperature, much like those field studies where a warming experiment, a warming treatment was imposed. And I think this is quite fascinating that we find all of the genotypes of Arabidopsis flower much later at the warmer treatment. And so this was a surprise to us, since the majority of data has indicated that warming might actually accelerate flowering time. This next slide will only show the effect of increasing CO2 over the last 100 years. In this case, we're not seeing any change in flowering time with higher CO2. So the warming is the same, it's still 20 degrees, but only CO2 has been risen. So what we're doing is decoupling the effects of rising CO2 and temperature in experiments that are changing together in the field, and we aren't seeing a CO2 effect. But if we have both CO2 rise and a temperature rise, which is this treatment, into the modern period, we actually finally then see the acceleration in flowering time. So what's driving this is a strong interaction between both rising CO2 and rising temperature, which accelerate growth rates but stabilize the size that these plants flower. And so in that case, we finally see that acceleration that we're seeing in the field. And therefore, it's likely that if these plants, if these model systems are similar to what's happening in the field, it might take both CO2 and temperature rise to see this accelerating effect. And that's going to be very important for us in the future as we try to predict what will happen with both rising CO2 and temperature. Now, to extend that a bit, we've actually taken the same genotypes and have grown them at very high CO2 expected for 50 years from now and much higher temperatures. And notice one genotype, the black genotype, can actually maintain the same flowering time across all of those treatments, but it's the only one. All other genotypes are doing very different things at future conditions. This is delaying its flowering relative to the current condition. Other genotypes are accelerating their flowering time. They're all over the place as we look out into the future. And likely not all of these will be winners. So in different environments, later flowering may be selected for, and in some environments, earlier flowering may be selected for. It would be environmentally dependent. So there we can see that it's critical that we actually disentangle some of the changing factors with climate, temperature and CO2 in this case, to really get mechanistic about what's driving changes in developmental timing between the pre-industrial period and the present. So in conclusion, just some broad points, and that is that climate and CO2 have changed over the Earth's history, and plants have clearly adapted to these changes. They always seem resilient, and they find a way to thrive even in conditions where it's dark for 4 months of the year, or in conditions where carbon dioxide is very low as to greatly limit physiology and growth. And I hope that I've convinced you that ancient plant collection are critically important for understanding the mechanisms behind how plants might adapt to CO2 and climate change. But the biggest concerning message is that plants don't have tens of thousands of years to adapt to the climate scenarios that we're looking at in the future. These will happen in 50 to 100 years from now. And the potential for plants to adapt that rapidly might be very problematic, particularly for long-lived species. And so it gives us a warning that contemporary climate change is possibly happening too fast for these adaptations to occur. So with that, I would like to thank the National Science Foundation, DOE, the USDA, as well as my home institution of the University of Kansas for funding this work. And thank you for your attention, and I will be happy to take any questions. Okay, sure thing. Hey, Janine.
Hey, so for your La Brea tar pit example with the juniper, do you think the fact that juniper being a gymnosperm, having evolved hundreds of millions of years ago when the CO2 concentration in the atmosphere was very, very high, that the rubisco in that tree would have had a different binding affinity for CO2, for example, than, say, an angiosperm that maybe evolved 10 million years ago, like a walnut or something, that might have had, Rubisco might have had a different binding efficiency or something, and you might have gotten different CI concentrations.
That's possible. We are actually planning to look at that in the Pack-Rettman collection, where we've been able to potentially isolate DNA. We're working on that now. That's a possibility. The general dogma is that Rubisco, as you well know, is fairly conserved over millions of years. And so I don't think that's as likely as the fact that it's a long-lived tree. It's adapted to very slow growth. It's adapted to shutting down during periods when resources are low. I think its life history traits have much more to do with its long-term survival in both high and low CO2 periods. I think that's more critical, possibly, than evolution of Rubisco. That's our thoughts on that at this point. Being able to just persist with slower growth is probably more important than the enzyme biochemistry. Sarah.
So the answer to this question might be related to what you just said. So I hadn't really thought before about the fact that these plants had to be in the dark for so much, in these really, really productive, hot, high CO2 environments. I don't know why that never occurred to me, but that's like really crazy to think about. And it seems like in that case that adaptations to slowing down respiratory rates would be like super important, and that might give us insight into species distributions in the future. And so can you just comment on what you think those adaptations might be?
I can kind of fill that in with kind of the thoughts on those samples that we looked at, the Glossopteras. So the idea at first was that they probably dropped their leaves prior to the dark season so that they wouldn't lose carbon in respiration. That was a long-term theory. I think it was proposed in the 20s or 30s. And then some work by Björling and a number of others looked at whether that made sense, and they found that it was more costly to drop leaves than to do more of an evergreen state. So the new idea is that the shape of those leaves and probably the photosynthetic machinery in those leaves was such that photosynthesis was maximized during light periods, and then they were able to store enough and then drop those leaves throughout the dark period. It still isn't quite clear what the advantage would be to dropping those leaves and having a deciduous nature because of the dark. It seems like more carbon would be lost, but they must adjust to that by having high photosynthetic capacity during light phases to have the storage capacity to make it through those 4 to 5 months of darkness. So I think the really critical issue is enhancing your storage capacity for that long term. Why there were not more trees there that would have saved their leaves for multiple years is not clear to me. I think that's still kind of in progress, but it's a good point. And as they move north in future climate change, I think, obviously, respiration is going to be a key issue. And the light regime might play some interaction with that.
Other questions?
Thanks for a great talk. I'm wondering about the phenology shifts that you saw, and if you have any speculation about why the plants are shifting towards later flowering times. I mean, I always expect, warmer temperatures, it's a longer growing season, so shifting earlier just makes sense to take advantage of that. But what advantage would there be to a later flowering?
So a couple responses there. So if you think about the physiology with high temperatures, as Sarah mentioned, respiration goes up under high temperatures, and so does photorespiration. So that's in cases where Rubisco is binding oxygen rather than carbon dioxide. So there are a number of physiological factors that can decrease plant growth at high temperature. We always think about that it spurs growth, but it doesn't always. Sometimes it hinders growth. And so this might be a case where lower growth rates due to heat, Arabidopsis is a cold-adapted plant, might make it more sensitive, and therefore its growth is reduced by the higher heat, making time to get to reproduction slower in that case. Now, the CO2 end is more interesting. We do see delays in flowering time at high CO2. That's harder to understand because carbon dioxide, if anything, would accelerate growth rates and they would get to flowering time faster. What we think is that the excess sugars that are produced at high carbon dioxide, we now know there's a sugar pathway for flowering time. And the plant is probably sensing those high sugars and for whatever reason are possibly delaying flowering time because of sensing of higher sugar levels. We are working and trying to understand that right now. And so we think that if anything, we don't think plants are sensing CO2 directly, but the sugar sensing is playing the main role on delayed flowering time. That's our hypothesis anyway.
Cool.
Is there any evidence in Arabidopsis that increased sugar levels in the leaves are having a feedback and decreasing photosynthetic rates of the leaves
Yes.
under elevated CO2? So that could also play into that?
That's possible. We do see, though, in all cases, we see at 700 ppm CO2 accelerations in growth, and different Arabidopsis genotypes have different levels of photosynthetic inhibition at high CO2. I think what you're getting at is, how does that relate to the sugar levels in flowering time? And if they downregulate photosynthesis and produce less sugars, what direction are we seeing in the flowering time? And I have a PhD student addressing that question at this point. So the idea is at 700 parts per million, and many plants will downregulate photosynthesis and actually have photosynthetic levels that are similar when grown at current CO2, and that may be affecting sugar levels. We do know that plants that delay flowering time at high CO2 have relatively higher sugars than those that do not delay. So it seems to be on the right track. There's a question.
You sort of answered it, but at what point in CO2 levels would you begin to get mortality absent temperature?
It's very high, several thousand parts per million. And that work was mainly done in regard to spacecraft, if we were growing plants going to space and we had very high CO2 levels. If I recall the studies, at several thousand ppm you start to see in corn banding on the leaves and so forth, but it takes high levels for there to be mortality due to a CO2 effect. Very, very high levels of CO2, if any, will do it. In some cases you can have damage from excess starch that can damage the leaves. But generally, if it's too high, they just won't use it. It usually does not cause plant mortality.
I was wondering if you could talk a little bit about what these different kinds of changes mean for us, or potentially mean for us living in the world. Why does it matter if flowering time changes or the growth rates change?
Okay. So getting back, it's a great question. Getting back to the surprise effects. So if suddenly we're banking on the fact that warming, let's say we assume warming is causing all of the accelerations in flowering time, we would model that and have predictions about how early a farmer might need to plant their field to have maximum gain on crops. But basic science tells us that there might be more to it. In my case, it might be a CO2 interaction with temperature that's driving that. And the more knowledge we have, the better models that we can build to predict flowering times that aid in knowing when to plant for crops, for maximizing crops, or for knowing when pollinators that are coming to visit flowers might become off sync and no longer have flowers to visit because the flowers came out too early before they were flying around to pollinate them, and to use those resources in the flower. Other areas, for example, the nutrient quality of vegetation changes at high carbon dioxide. It actually has less nutrients relative to carbohydrates. That could affect human health. There are a number of reasons why, and not to mention the fact that plants, like I mentioned, are that energy carrier where the sun's energy is put into the ecosystem through plants. It's our food source. And so any impacts on plants in an ecosystem have very far-reaching consequences for us.
All right, and with that, I think we should thank Dr. Ward once
you.
more. Present you with our lovely parting gift.
Oh, wonderful. Thank you.
Frontier's mug.
Appreciate it.
There are some snacks on the back, and I encourage you all to stick around and chat. Next week, Larry Wackett, INE Fellow and Distinguished Knight University Professor, will take on the question of how humans can emulate nature and recycle everything. So thank you all for coming.
Thanks very much for having me. I really enjoyed it a lot.
I'm Louis Gilbert. I did never.
Nice to meet you.
Appreciate your patience with the,
Oh,
with
no,
the
I
technology.
should, yeah,
My guess we're trying out new systems, so there's probably something funny.
yeah. I think it's something kind of connection. I don't
Oh.
know. Who knows?
Well,
Anyways, fun. Thank
glad
you.
you could come.
Yeah, I am too. Yeah, I know. Really good to see you.